morning, all. Thanks for coming early. So yesterday we hit a lot of modeling. Today I think you're going to hear more research. And our first research panel is going to go over contaminants of both emerging and growing concern in the Bay watershed. So the way we'll be set up, I'll moderate the session. I coordinate the USGS science efforts for the Bay program, and I also admit the Bay program a couple days a week to help coordinate the partner science efforts, working along with Rich. So we have Greg Allen here also. Greg coordinates the Toxic Contaminant Work Group. And then we have three speakers that I'll introduce later on. And I'll give a little intro, and then we'll have our three speakers. Then we'll all come up as a panel, and you guys can ask some more direct questions and have a discussion on toxic contaminants. Wanted to thank Bill Ball. He helped put this session together. And I have a trivia question for you. So 3 decades ago, who was the toxic contaminant coordinator for the Chesapeake Bay program? Any guesses?
Rich Batuk.
Rich Batuk, you got it. So Rich started this effort 3 decades ago. He's moved on to what you heard yesterday, but we still have a ways to go. But Rich, another one of his legacies of getting this started. So thanks, Rich. So why are we talking about toxic contaminants? Well, there's several big concerns in the watershed. One, they're a threat to human health through fish consumption advisories. We've had a lot of evidence of degraded fish and wildlife from toxic contaminants, and they're widespread throughout the watershed. And here's a little bit on what we know about their occurrence and severity. If you look at the tidal waters, and that's the map here, 80% of the segments in the bay are either fully or partially impaired due to toxic contaminants. PCBs, mercury, and pesticides are widespread throughout the watershed. And then we have these emerging contaminants that have widespread use, such as pharmaceuticals, care products, flame retardants. But we don't have the monitoring of science to say whether they're actually widespread occurrence. And that's a lot of what we'll talk about today. As far as impacts, I mentioned fish consumption advisories. Within the bay, PCBs are one of the main fish consumption advisory causes. And if you look at this striped bass here, let me get this set. One of our iconic species, but you only can eat about 8 ounces a month due to the high PCB levels. Up in the watershed, mercury is the main contaminant causing fish consumption advisory. So while we don't deal with human health effects in our work group, we do deal with the issue of fish consumption. We have a lot of examples of degraded fish health. If you look in some of the fishery species, we'll see lesions and tumors. We also have reduced reproduction. We have intersex conditions or feminization of fish. And we feel like toxic contaminants has also reduced the immune function of fish and made them more susceptible to infections and other parasites. And you'll hear about some of these issues today. In wildlife, most of the issues have dealt with water birds, where we've had eggshell thinning, actually embryos dying off due to pesticides, and then hatching success due to legacy contaminants such as PCBs. So because of all that, the Bay program, in their latest watershed agreement, had toxic contaminants as one of their 10 goals. And this is the aspirational goal they set up for contaminants. And under that goal, there's 2 outcomes, one on policy and prevention, and the other one on research. And we'll be talking mostly about the research outcome today. But we work on those as a partnership through the Toxic Contaminant Work Group. And we just went through our first set of plans and work plans, and we're going to revise those. So there's a real opportunity to get more involved, to bring the research forward, such as you'll hear about today, and other partners to try to address some of our big science gaps. So we really need and welcome more involvement from the research and academic community on this issue. These are our 5 research objectives we have in the toxic contaminant work group. One is just trying to provide the science so jurisdictions can make fish and shellfish safer for human consumption. We want to better understand, we know there are effects on fish and wildlife, but which contaminants might be causing those effects? It's a very difficult issue to get towards. When we can understand which contaminants might be causing the effects, really better understand their occurrence, sources, and concentrations, so we can come up with options of how do you reduce their impacts. And then we know there's always going to be issues of emerging concern. Last year we did a synthesis on microplastics, so as new things come up, we're looking forward. So the talks we have today will cover all aspects of these different research objectives. And about 5 years ago, we did a summary report of what do we know about toxic contaminants. And these are the contaminant groups we looked at. And we tried to better understand what we know about currents, concentration, sources, and effects. And this just gives you a snapshot of our qualitative assessment of where we think we have enough information in green to move forward on a management strategy, and where we're really lacking information in red and yellow, where we need to improve our understanding so we can better assess how to reduce their impacts. So a lot of what you'll hear about today deal with these issues here. We took this information, and the framework we move forward of working together between the research outcome and the policy and prevention is this, where we have enough information to try to move things forward, such as PCBs, we've developed a policy and prevention outcome. And Greg Allen will talk about that briefly during the panel discussion and what some of the remaining science needs are for that. And also, we know we need to tackle mercury, given how widespread it is, but there's more information needed there. These were both very widespread. Now, where contaminants were sort of local effects, we decided we're not going to tackle that as a Bay program partnership. We're going to let the states deal with that in their localities through the TMDL process, because we're trying to look at things that are widespread. And then this last grouping of contaminants, these were sort of the red category in my previous slide. This is where we developed a research agenda to better understand these aspects of it. So that's sort of the logic we used. And as we get more understanding of these, we can bring these up and start to develop policy and prevention strategies. So, final slide. I think here's the opportunities and some of what we're going to hear about in the panel today. So, we need more information on these different aspects, biological effects and their causes, what the occurrence and sources are, and then the management implications. And I think one of the things we really need to try to work on is how can we take advantage of all the work being done on nutrient and sediment reduction that you heard about yesterday, and also reduce contaminants. And the reason I say that, many of the sources are the same, whether it's agricultural areas, air emissions, runoff from industrial areas, wastewater treatment plants, or suburban areas. So we have the same sources. In general, it's the same pathways. They're either moving through water or moving with sediment. So as we put in practices to control things in those different landscapes, we want more research to say, here's a benefit we can also get for these PCBs or other emerging contaminants that we're trying to control. Because we don't see ourselves being able to do a whole separate management effort toward contaminants. We really need to take advantage of the big TMDL effort that you heard about yesterday. So with that, I'm going to bring up our first speaker, Vicki Blazer. And so Vicki's a fish pathologist, researcher at the USGS lab in Leetown, West Virginia. She's done work internationally. Well, we're blessed to have her here in the Chesapeake. Next week, she's up in the Great Lakes talking about results. She's been across the world, and she's also an adjunct professor at West Virginia University and Penn State. And many of her students will have posters here last night and tonight, and there's a session tomorrow on, I think, bioindicators. So, Vicki.
Thanks. Okay, so in the aquatic environment, there are obviously thousands of compounds that are being introduced all the time. And these are in complex mixtures of both legacy and these contaminants we call contaminants of emerging concern or emerging and increasing concern. And these all vary temporally and spatially depending on climate, source, hydrology, habitat. And so aquatic organisms are constantly exposed to these numerous contaminants through many pathways, through the water, through sediment, through food, and sometimes through the egg. Some examples of some of these that we're beginning to look at, that, as I say there, they're called contaminants of emerging concern, but really we've been looking at them for quite a few years. Pharmaceuticals that come from both human and veterinary sources. Hormones, again, both human and animal, agricultural sources. Phytoestrogens is something that we only recently began looking at in terms of some of the fish health issues. Current use pesticides and herbicides, chemicals in personal care products, so those include things like the UV blockers and sunscreen, antimicrobials, microplastics, all those sorts of things. And of course, sources are also many. A lot of things have been linked to wastewater treatment plants or other sources of sewage, but we also know that agriculture can be a big source as well. So manufacturing plants would be an example of a point source, but a lot of our things I think come from non-point sources. So some of the examples of the kinds of things that we're seeing due to some of these emerging contaminants, probably the one that's attracted the most attention and has been looked at worldwide is this intersex issue, or what we call it testicular oocytes, so basically immature eggs within the testes of a male fish. But we also see effects on female fish as well. Inhibition of reproduction, as well as masculinization of females. As far as antibiotics, everybody probably knows about antibiotic resistance, but one of the things that we're really concerned about is how some of these antibiotics and antimicrobials are affecting the normal flora. Most of you have heard probably about the gut biome, but fish on their surface also have a very important biome in that mucus covering that is their first defense. And so these things can really affect that. I mentioned metformin just as one of the pharmaceuticals recently that I certainly had not thought about as a potential cause for intersex until just recently, where it came out from some work in Milwaukee where they found it correlated with intersex and then went back into the lab and indeed induced intersex with metformin. Metformin is probably one of the most prescribed pharmaceuticals for diabetics as well as polycystic ovary. And so it is one we had not been looking for up until recently, and when we started looking, we actually are finding it in a lot of our water samples. The other thing is antidepressants. Again, apparently well prescribed, and we actually can measure antidepressants in the brains of fish. So we did a study in Iowa a number of years ago and actually measured 5 different antidepressants in the fish downstream from a wastewater treatment plant. So some of the complexities that you have to think about with these is that unlike our legacy contaminants like PCBs and mercury, these potential contaminants are produced to have a biological effect. And so they can have a biological effect on fish and other organisms in the aquatic environment at very, very low levels. And while fish have a lot of the same systems that we do, and so we can use a lot of the same methods that human medicine does, we really have very little understanding of how some of these things affect invertebrates and other lower organisms that are a really important part of the food chain and that we need to be looking at. The other thing that makes it somewhat hard to understand some of these things is that for the endocrine system as well as for the immune system, they're regulated through chemical communication and feedback mechanisms. And so very often we don't see the nice dose-response curves that we do with, say, PCBs or mercury. The other thing is that we know, again, for both the endocrine system and the immune system, is that exposures at a young age, the most sensitive age, can have really long-term effects that may not show up until they go through sexual maturation. And then the other complexity is the mixtures. So when you have estrogens and androgens, do they somehow antagonize each other? We don't really know. So we've been using resident fish as integrators for this aquatic health system because they're constantly exposed, they integrate over time, and they integrate the various stressors, whether that's increased water temperature, other climatic effects, infectious agents, etc. And so while they are good indicators, it also makes it really difficult to tease out actual causes or risk factors. So, as Scott mentioned, some of the things that we have seen here in the Chesapeake, we certainly see the reproductive endocrine disruption in centrarchid fish. We see intersex. In yellow perch and some of the urban tributaries, we're seeing a lack of reproductive success. And I'll talk a little bit about what we see there. And then there have been population declines of numerous species that may or may not be associated with some of these contaminants. The other thing we do see is neoplasia or cancer, and a number of these emerging contaminants have been associated in warm-blooded animals with cancer. And so we're asking the questions, is that one of the risk factors in fish? Here in the Chesapeake, we see both skin and liver tumors in bullheads and, for instance, the Anacostia and some of the other urban tributaries. We've also seen testicular tumors in yellow perch. And then one of the things that we're really interested in is this immune modulation, because we do see a lot of skin lesions and fish kills. We also see fish that have multiple co-infections of opportunistic bacteria and viruses. And so that raises the question, why? Why are they getting these infections? And then heavy parasite loads as well. So as I said, we have primarily focused in our group on biological monitoring, where we're looking for adverse effects and using what EPA has termed adverse outcome pathways. So we actually are looking at a whole range of biomarkers or indicators from the organism level down to the gene level. We, for many years, have done the organism level and the tissue as far as histopathology, which is what I love to do. We've recently added a lot of immune function as well as the molecular. So we're looking at sets of genes to try to tease out what are the mechanisms for some of these things that we're seeing. So some of the things we've learned, all fish don't respond the same way, and so it's really important to pick a fish that is going to respond to whatever it is you want to look at. So species choice as well as choice of biomarkers is really important, and I'll give you some examples of that. The other thing I think I've come to the conclusion is that we're not going to find one cause. Numerous chemicals can have the same effect, for instance, in intersex. So for instance, in an early study we did, we call our spring spawning study, where we looked at 7 sites in the Potomac, and we had water contaminant analysis done. The only things that correlated with intersex were the herbicides, particularly atrazine and metolachlor. On the other hand, when we did a similar study up in Susquehanna, there we had 11 sites, and that was done in the summer instead of the spring. The only contaminant that really correlated was estrone. So again, correlation is obviously not cause and effect, but it does give us some examples of things that we might want to look at in the lab or in more detail. The other thing I'll point out is the study that we did up in Pennsylvania, three different watersheds. We looked at white sucker at the same places we collected bass and red horse sucker in the Ohio drainage, and in no cases did we see intersects in the suckers. So we see intersects in bass, but we don't see it in the suckers. And we've seen a similar thing up in the Great Lakes as well. But what we did see in the suckers was vitellogenin in males. So there are numerous ways we can look at exposure to estrogenic compounds. Intersex is most likely induced early in life, but can increase in severity as a fish is exposed. Vitellogenin in male fish is something we can measure non-lethally in the plasma or the blood, and so that is probably more indicative of more recent exposures, days to months. And then we're also looking at gene expression of vitellogenin, which would be more indicative of a much more recent exposure. In the yellow perch, we see something very different. We don't see intersects in the yellow perch. We do see some effects on the male fish. We saw a couple Leydig cell tumors and proliferation. But what was really obvious was the effect on the females. And let me see if I can pointer. So a normal egg has nice yolk and this very thick eggshell around it for yellow perch. What we were finding in the tributaries where we don't get good reproduction is abnormal yolk, and you can see this really thin, abnormal egg covering or eggshell. So in this case, it appeared to be the females that were primarily affected. As I mentioned, we do see tumors in fish. So in bullheads, we see melanomas, squamous cell carcinomas, papillomas, and liver tumors. Interestingly enough, again, up in the Great Lakes at sites, and we did multiple sites where we were looking at both either bullhead or white sucker or bass, we rarely see tumors in bass. So again, differences in species. So basically what we're trying to do is we look at the biological effects, but working with others that are looking at chemical monitoring in both the water, sediment, and hopefully fish tissue eventually, including in-depth land use and land cover, and that hopefully will give us an idea of what we should be looking at as far as laboratory exposures to prove some of these things. The other thing I didn't mention that I probably should have put in, especially for this group, is as we get all that huge amount of data together, which we will have, modelers will be real important to help us understand what this all means. And that's what I have. I just want to acknowledge that there have been many groups and organisms involved, not organisms, organizations and organisms involved in this research, and a number of graduate students from both West Virginia and Penn State. Thank you.
So Lee's an associate professor over at UMBC, and he looks at basic contaminants, emerging contaminants in natural and degraded environments, and also tries to focus on recovery from agricultural waste. So Lee, why don't you come on up and share some of your thoughts with us.
Good morning, everyone. Good morning. All right, some of you know the UMBC thing. Our president always says, good morning, doesn't get a good response, and then says, good morning again. So good morning again.
Good morning.
Good morning. Trying to learn from my leader here. I'm really happy to be here. I've been at UMBC for about 7 years now, but this is actually my first time at this meeting. Yesterday was really great. I learned a lot. I particularly liked the oyster session yesterday afternoon. We've been working with oysters. So it was really cool to learn a little bit more about them rather than just measuring the chemicals that accumulate in them. But yeah, so let me tell you a little bit about what we're doing. First though, I do want to acknowledge all of our collaborators and funding sources. On this particular work, the data that I'm showing, The underlined people here, Ka, Ann, Mitch, Ethan, and Anna, we're all very critical in collecting these data, either helping us kick off the sampling campaigns or actually processing the samples themselves. The other individuals listed here really helped out with the sampling campaigns, and then the funding was mostly through Sea Grant, NSF, and the USDA Farm Service. Okay, so one of the things that I think comes up often with contaminants of emerging concern, or CECs, is that there's so many chemicals. Where do you even start? Which ones do you measure for? In the FDA, I think the FDA regulates something like 4,500 different pharmaceuticals. So when you think about those pharmaceuticals and then the potential metabolic products and then the potential environmental transformation products, we could easily be thinking about 10,000 or more different chemicals. And so where do you start? We really wanted to start by thinking about those classes of compounds that in our mind, so the little star on priority here, that's just our priority. We're not trying to speak for anyone else. These are the ones that really, I think, have a clear concern with respect to either public health or ecological health. I was very glad to get to go after Vicki because she gave a great intro on some of these things, and so maybe I'll go a little bit faster through here. Antibiotics, our big concern is definitely the development and spread of antibiotic resistance. If we're loading antibiotics into the natural environment, that is probably not helping our case with respect to antibiotic resistance, although there are several other very important mechanisms there, thinking about overprescription and thinking about these things. I'll just note ciprofloxacin. This is an antibiotic that we work with quite a bit. In 2000, this map was all the lighter orange color. The darker red colors here indicate the presence of resistant E. coli to Cipro. And so even in one decade, we've seen very major changes in resistance patterns throughout the US. I think right now antibiotic resistance costs us about $30 billion every year in this country, and by 2050 is going to be the leading cause of death. And so what do we do when antibiotics stop working? I don't know, that's a big question, and putting these things into the environment probably isn't helping. Estrogenic hormones, which Vicki talked about quite a bit, we're concerned with some of those intersex issues, other endocrine disrupting effects. And importantly, these can have those impacts at very low concentrations that are relevant to what we actually see in the environment. Less than 10 nanograms per liter for sure, for some molecules down closer towards 1 nanogram per liter. And they can have devastating consequences for different organisms in different areas. And then the UV filters or the sunscreens, which Vicki also mentioned. We're really interested in these. These are present in a lot of personal care products. If you have something that has an SPF number listed on the front, if you look on the back side, you see a bunch of organic chemicals. We're probably measuring for all of those right now. These have weak estrogenic activity, and so they can also cause some of that endocrine disruption effects. But most recently, these were in the news because 2 of them were just banned from being sold in Hawaii. Oxybenzone, or BP3, and ethylhexylmethoxycinnamate, or EHMC, which is also known as octinoxate. Those 2 are definitely ones that we measure, but we really measure all of the FDA-regulated UV filters. So there's about 17 of those. And I'll just note this picture here. I hope you can see it from the back. This is a photo that's not from Hawaii, it's from Central America, but it was a diver that had put sunscreen on, protected themselves, went down and touched this coral. And what I hope you can see is the photo bleaching of the handprint just from that. And so the concerns with these compounds really are thinking about toxicity. Corals obviously play a very important role in the marine ecosystem, and so if we're kind of exacerbating other effects of climate change through the discharge of these chemicals, again, probably not a good thing. In total right now, we're measuring about 40 to 50 antibiotics in environmental samples, about 15 hormones, and about 15 or so UV filters. So around 70 or 80 different compounds. I'm not going to get into the methodologies here today, but if you're interested, I'm happy to follow up after. So we do a couple of different things in the Bay. I consider myself an environmental engineer first and foremost, and so we kind of started out thinking about wastewater treatment processes, drinking water treatment processes. How can we take some of these CECs and transform them through chemical or photochemical processes into other chemicals that may not have the same concerns? Okay, and that's a really important aspect there. One of the reasons that we focus on particular classes is that one of our guiding hypotheses is if I have an antibiotic and I'm treating it and causing some structural change and treating it and causing some structural change, some of those products I'm forming may also be antibiotics. Indeed, we see that occur in wastewater treatment plants. We see that occur in the natural environment. And so again, the number of compounds is really important because if we're just looking at one and we're seeing the concentration go down, that doesn't necessarily mean that we've removed the concern. Those transformation products can be equally potent. They can actually be more potent for some of the antibiotics that we work with. We also have done some work in the Glens Falls watershed. This is part of a long-term project that we're working on for the next 4 or 5 years. This is a really interesting watershed for us because there's no wastewater inputs. There's no big animal feeding operations adjacent to this watershed. And so we shouldn't expect to find the CECs that we're looking for in that water, but we do. And so we think that's coming from leaking sewer systems. And these things get into the water, they get into the sediment, and we've done some work showing accumulation in crayfish, including the chemical ethinyl estradiol, which is the birth control drug. So these things do accumulate in these urban organisms. They are very potent estrogens, and so I think we can see some impacts there. Okay, so I'm not going to talk about one or two here today. I just want to give you a little bit of an intro there. If you're interested in any of that work, I'd love to continue the conversation later. But for today, I really want to focus in on some of the stuff that we're doing really in the main part of the bay in Maryland. Okay, so we started this work pretty recently, actually. The first time we went out was summer of 2016. And so we went out with a couple of guys from the Maryland Department of Environment up to the Chester River. They were doing some other things up there. We kind of just wanted to tag along, collect a couple of samples, and see what we found. So very much exploratory. And so what you can see here, these are water phase concentrations of antibiotics. There's really two that were predominantly present. In the orange is sulfamethoxazole, and in the blue is norfloxacin. These are both human use antibiotics. And we were really surprised by these results. These are actually pretty high concentrations. This is upwards of like 100 nanograms per liter. And for norfloxacin, that's where we can start to select for antibiotic-resistant bacteria. Okay. We're in a pretty open area of the Chester River here, kind of right where it discharges. So large body of water, a lot of dilution, a lot of mixing. We were not expecting to see these concentrations, especially since the upstream population is pretty small. And so this was kind of eye-opening for us. Yeah, actually, maybe we do have an environment where we can select for resistance, in the main part of the bay. And so we went back out to these sites that we're going to call the Lower Eastern Shore area. This is in Maryland, near Princess Anne, right up here. And this was in conjunction with the Maryland Department of Natural Resources. We first went out in the spring of 2017. I'll show you some results from fall of 2017 a little bit later. Okay. What I hope you can see here is coming down through the Minokan River, we see this kind of gradient of antibiotics being discharged. In the purple, green, and orange, these are all macrolide antibiotics, which are available for certain uses in animals, but are predominantly used in humans. The one that I'm really interested in is this yellow one right here. This is a chemical called enrofloxacin that is only supposed to be used in animals. And so kind of looking at these results in tandem, we saw some human use antibiotics, we saw some animal use antibiotics. And so we're really interested in kind of building out this data set to get a better sense of, okay, are these things actually coming from wastewater? Are they coming from animal feeding operations? Can we track that? And can we kind of start thinking about, the potential source, regulations to limit these things getting into the bay? The other thing I'll note here, if you can't see it, there's a couple of sites that we looked at right here in Holland Straits. This is pretty close to the central channel of the bay. And down here in Kids Creek, this is pretty isolated coastal bay. And we didn't find any antibiotics here. So that was kind of a good check for ourselves. We're not seeing these things in places where we don't necessarily expect to. But when we switched over and started looking at the UV filters and the estrogenic hormones, we did find them in Holland Straits water. We did find them in Kids Creek water. I'm sure some of the modelers in the room can help us, get a better sense of where these things are coming from. Are they coming from the across the bay? Are they coming from upstream? But we find pretty sizable concentrations of this slew of UV filters at these sites. And again, we kind of see this nice gradient coming down through the Minokan River. These chemicals, the UV filters and the hormones, are much more hydrophobic than the antibiotics. And so we're also interested in whether or not they're accumulating in the sediment, whether or not they're accumulating in oyster tissue. And so we looked at those phases as well. For the sediment, we see a pretty similar picture here. The blue bars are a molecule called homosalate. This is the compound that is used in the highest percentage in sunscreen products. It can be as high as 15% of a sunscreen as homosalate. And so unsurprisingly, we find a lot of that here. And then the purple and the yellow at the most upstream Minokan site, that is estrone or E1 and estradiol or E2. These are potent estrogens, which again is kind of saying, yeah, we definitely have a source of these molecules coming down the Minokan River that bears more looking into. And then for the oysters, we see a kind of similar picture. The distribution of molecules changes compared to the sediment, but again, we see concentrations of UV filters through the Minokan, out in Holland Straits, and down in Kitts Creek. And the one thing I'll note here, the red bars and the gold bars, those are the two UV filters that were recently banned from sale in Hawaii. And so these chemicals are definitely going into the water phase, they're getting into the sediment, and they're also accumulating in the oyster tissue. What are the concerns of that? We don't know in oysters, but as I mentioned, there are definitely concerns that have led to regulation in other organisms. And so we were thinking about these results and we said, okay, well, this is kind of a limited number of sites. We really want to expand this out. And so talking to the guys at Maryland's DNR, especially Mitch Tarnowski, we learned about their annual oyster surveys in the fall at many more sites. And so we worked with them to collect water, sediment, and oyster tissues from around 55 sites across the bay last fall. Okay, so here I'm showing just the UV filters. It gets like way too crowded otherwise. And so this is 8 UV filters that were detected at these sites all throughout the Maryland part of the bay. Again, homosalate, which is used in the highest concentration, was the most frequently detected at the highest concentrations in the blue bars here. There hasn't been much work with this molecule, but based on the structure, we can expect to see some estrogenic activity, perhaps, and we have backed that up with some lab studies. And so this is one that a lot of people haven't really looked at as yet. There's some analytical complexities, but we're definitely finding it everywhere, and it's kind of as expected based on consumption at high concentrations. I'll also just note here the red symbols, which I hope you can see, those are wastewater treatment plants. And so we definitely have larger wastewater treatment plants on the western side of the bay. And then the blue, these are all just kind of a little bit of an indication of agricultural activity. So these are poultry feeding operations. And you can see, as probably everyone here knows, there's a high concentration on the eastern side. We can break these results down by river system. Right now we just have that one time point from fall of 2017. And so I think there's probably a lot of uncertainty in these data, in terms of daily changes in concentration, weekly changes in concentration, things like this. But there are some takeaways that we can pull from here. So first of all, the Potomac River, we see the highest concentrations of these UV filters coming through. Given the upstream activity, given the upstream load of wastewater effluent, we think that makes sense. And then in the Choptank River, we're seeing a bit of a different distribution of compounds. We're not seeing the octasalate and the gold that we do everywhere else. And also, it's kind of interesting for us, the ethyl hexyl methoxycinnamate, the pink bars here, we're really only seeing that popping up on the eastern side of the bay. That may be due to differences in wastewater treatment train. That's something that we're looking into more right now. On the oyster side of things, again, I'm just going to show the UV filters here so that I don't kind of overwhelm. And we're still waiting on data from about 10 or 15 of the sites, and so this is about 40 of the 55 that I'm showing you. Again, the point that I really want to make is that these things are showing up in oyster tissue from around the bay, and especially homosalate and octasalate, which is a very similar structure. In homosalate, this alkyl group kind of swings around to form a ring. Okay, but again, we kind of have this phenol moiety in octosalate, which can demonstrate some estrogenic activity. If we look at the distribution of UV filters, the concentration of UV filters in the spring and the fall in water samples and oyster samples, we can also see some interesting things. So on the left-hand side here, I have the water samples, and then I have 3 sites. So it goes spring, fall, spring, fall, spring, fall. What I hope you can see here is that the concentrations are pretty similar in the water phase between the spring and the fall. I will note that the gold, the octosalate, was not measured in the spring, and so you can kind of ignore that one for comparison purposes. Otherwise, these line up pretty well. In the oysters, we see a very different story. We're seeing much higher concentrations in the oysters that were collected in the spring, and we're also seeing this different distribution where we have way more oxybenzone and ethylhexylmethoxycinnamate, again, those 2 sunscreens that are regulated now in Hawaii, present in oysters that were collected in the spring. I will note that these oysters were different ages, different sizes, and so there's a temporal aspect in terms of the loading and the concentrations of these compounds, but then also the accumulation of these molecules within the oyster tissue itself, which again, we want to collect more data so we can get a better handle on some of these kinetic aspects. So I think I'm probably running out of time here. And so in terms of my concluding thoughts and research needs, we use a lot of sunscreen. We think sunscreen is important. We want people to protect themselves, protect their skin. But thinking about ways that we can kind of start reducing the load of these molecules getting into the bay, we think is quite important. We want to get better data on some of the spatiotemporal trends, the sources, the mass loadings, and then also biomagnification. We've only looked at some mussels and eastern oysters. Looking at other species is something that we're interested in. So if you have other samples, if you have other things, we would love to keep that conversation going. Wastewater upgrades, best management practices for limiting nutrients and sediment, as Scott mentioned. We think there are probably co-benefits for reducing the loads of CECs into the bay. This is an area that I think is very much understudied, and I think it would be an opportunity again to kind of bridge the gap between some of the CEC work that we're doing and some of the nutrient sediment work that some of you may be doing. And then finally, these molecules were designed to elicit a biological response. That's the reason that they were created. And so when we're putting these molecules out into the environment, making sure that we're measuring the chemical concentrations, but then also considering the biological response is quite important. Otherwise, we can have a lot of data. We can see detections of everything with more powerful instruments or different sample sizes. But unless we're really coupling up to some of the toxicity work, it's hard to draw conclusions. And so again, that's an area where we would be very much interested in working with some of you to help draw some conclusions on what these concentrations actually mean. So with that, thank you very much, and I look forward to the time.
So Heather's an assistant professor at Penn State, and her research seeks to understand the factors that affect the presence, fate, transport, and ecological impacts of contaminants of emerging concerns. So, Heather.
Hi everyone, good morning. Thank you so much to the session organizers for first creating this panel and of course for inviting all 3 of us to be part of it. It's really an honor to get to speak in front of all of you today, so thank you again. And so I've been at Penn State for about 5 years now. I got hired initially to do emerging contaminants work, and I'm now starting to do a bit more related to the Chesapeake Bay watershed, so this is a great audience to be in front of today, so thank you again for this opportunity. But my approach has always been to look at the field scale processes of the fate and transport of these contaminants, and I've collaborated with a lot of ecotoxicologists along the way to try to make sure that the data that we're generating in the field is actually used to understand what the effects might be. So that's sort of the overall approach that I take in my research. The 2 main sources of these contaminants that I generally think about are concentrated animal feeding operations. So we've already heard a lot about veterinary drugs today, as well as the importance of manure as a hormone source. And then more recently, I've been starting to think about human sources as well. So I went to grad school in the Midwest and can't do water quality in Indiana without thinking about agricultural activities and their impacts on water quality. So I focused primarily on CAFOs early on in my research career, and I'm now, as an environmental engineering background, in an ag and bioengineering department, trying to bridge sort of these interests and the occurrence of these compounds as they're linked back to both human and animal sources. And so overall, as I mentioned, I take a field scale approach to research. So I guess what that means is that I tend to think about what hydrologic flow pathways are moving these contaminants in the environment. And so we hear a lot about hot spots and the spatial distribution of these compounds, but one of the things that I like to do is try to understand the moments in time that are also hot. So thinking about sampling over multiple rainfall events, over multiple seasons, and then understanding those temporal dynamics. Because if you do, especially thinking about manure application, if we're doing fall versus spring application, when the peaks of these compounds that are likely to cause impacts on spawning fish might be. And so maybe you get different effects ecologically if you do spring versus fall manure application. So thinking about the importance of these hot moments in time and what that means for ecological health. And then, of course, we want to understand the presence of these contaminants in surface water bodies like our river systems and our drinking water supplies as well. But one of the things that I've tried to do over my time at Penn State is understand their presence and sort of, I like to think about emerging contaminants in emerging ecosystems. So I know vernal pools are not an emerging ecosystem, but nobody's really looking at the presence of these contaminants in wetlands, both, wetlands that don't dry out in the summer, but then these vernal pools, which do have a hydro period that allows them to dry out in the summer and then fill back up in the fall, and they tend to have a very important habitat for, like a functional habitat for frogs. And so thinking about the impacts of these kinds of contaminants and our wastewater activities and septic tanks as a potential source of impacts to vernal pools. So I guess that's another one of the systems that I'm studying. And then trying to move, you've heard a lot today that would instill fear, right? I'm sure a lot of you are like, oh my goodness, what are we going to do about all these problems? We don't know all of this stuff. We have all of these ubiquitous sources of these contaminants in the environment. But how do we move from fear to empowerment? So how do we get people involved in actually solving this problem? And it turns out we have a really great citizen science eagerness across the Bay watershed. And probably a lot of you are aware of that. But people know about the fact that pharmaceuticals are in their water. They know that it's getting into their drinking water. And instead of just making people feel afraid about that, what can we do to actually enable them to make a difference? We're not going to have regulations anytime soon. We're not going to upgrade every wastewater treatment plant overnight. So what can we do as individuals to make a difference in this problem? So hopefully I'll touch on each one of those general approaches to my research today so you get a flavor for the kinds of things that I do. So one of the first things that I did when I got to Penn State was looked around for existing field sites that I could leverage. And right near the university are about 12 plots that receive manure application, and they're tile drained, so they have a subsurface and a surface runoff component. And 6 of them receive dairy manure via shallow disc injection, so that's putting the dairy manure about a few centimeters under the soil surface so that you reduce the amount of odor emissions that you have during the time of application and other benefits for doing manure application in that method. And then comparing that to the regular surface broadcasting method for applying manure. And so we have these huts sort of at the bottom of the site here that collect flow via surface runoff and subsurface flow from each of the plots. And then all of that flow goes into these little tipping bucket mechanisms. So if any of you know how a tipping bucket rain gauge works, it's the same thing, just larger. And so basically, you know the flow volume in each side of that tipping bucket. And so every time it tips, you would know the volume of flow that's passed through your system. And so that enables you to do flow-paced samples so that you can get an idea of what the chemograph changes are over the flow event if you discretize those samples, or if you put them into a composite sample, then you're getting a fluid composite sample over the event. And so we did that. My first PhD student did that work to look at the fate and transport of these natural hormones that are present in dairy manure. And then basically tried to sample as many events as we could until we didn't get any more surface runoff events the following summer, following an October 2nd application back in 2014. And so we were able to collect samples from a total of 10 runoff events. And the kinds of things that we were interested in understanding were first flush effects. and differences between the two different manure application types. I guess the contribution of snowmelt effects. So a lot of people take out their monitoring equipment over the winter because keeping it out in frozen conditions is very problematic. You have to have good field technicians to help you make sure that your equipment is working through the winter. But what would we be missing if we didn't capture those events? So trying to understand the importance of those snowmelt conditions. And then of course, spring rainfall. We had this kind of interesting hail event during our study period too as well. So those were the kinds of events that we were able to sample across. And what you're seeing here is the cumulative export. So basically the total loads as they increased over time following the manure application. That's kind of shown in red. So this red dashed line is that October 2nd manure application. Turns out that year, 2 days later after the manure application, we had a relatively small event. So you'll see it was actually the smallest rainfall event out of all the events we sampled. And it generated quite a bit of surface runoff from only the plots that had received the surface broadcast manure application. So that's when the manure just gets applied to the soil surface. It actually generated surface runoff from those plots that led to the export of 90% of our entire sampling period. So this 9-month sampling period was when the estrogens got exported from those specific plots that had received the surface broadcast. So basically this huge first flush effect. And none of the other events that's during that sampling period for the next 9 months really contributed much to the total load. Whereas in the shallow disk plots, we actually didn't even get runoff from the sites that had received the shallow disk injection. So just the difference in the timing of when the estrogens got exported following that manure application. And so we saw this huge first flush effect for the surface broadcasting and not for the shallow disk injection sites. But then the remainder of the time, so this blue is the surface runoff, and then red is total dissolved phosphorus, and yellow is dissolved organic carbon. So we're starting to think about differences or similarities in the transport of nutrients and hormones. So we've heard the other two panelists speak about that. What can we learn about nutrients? We know a lot about pathways for nutrients, and how can we use that information to understand what's similar and different about hormones, in this case, in the environment, and what differences in management practices like manure application techniques would cause differences in the fate and transport of those compounds. So we started to do that a bit. You can see for the shallow disk sites, the compounds primarily all track the surface runoff, and that was not so much the case for the surface broadcast. We saw this huge flush of estrogens, but not so much for the other compounds, and the nutrients more track the surface runoff. So if you're interested, I have other thoughts on why we saw that, but you can ask me during the panel or after lunch or whatever later. And so these were the sort of take-home messages of the study, that we saw the importance of the first flush for the transport of hormones from surface broadcast plots. We didn't see that for the nutrients or for the hormones from the broadcast. I'm sorry. We did see, we did not see the first flush effects for nutrients from the surface broadcast plots, only for the estrogens. And then we saw that the nutrients tracked both surface runoff for both application methods. And of course, we also saw the importance of snowmelt. So these events were like all snowmelt events, and if we hadn't monitored them, then we would have missed the transport of contaminants during that snowmelt period. So these were the benefits we were able to document. So now we have to think about, okay, most farmers are not doing shallow disk injection, so how do we get them to adopt a practice that that benefits compounds that aren't regulated? So you have to tie that back to nutrients, plant yield, and try to convince them that it's worthwhile to spend the extra money to have the shallow disk injection equipment, to spend the extra time getting their fields ready for manure application and harvesting, all these other things, and then try to promote the other known benefits of the shallow disk injection method in order to actually then make a difference, because you're not going to convince a farmer to change a management practice because of hormones. Or not likely to anyway. So another, research site that I've been doing research at over the past few years at Penn State is Penn State's Living Filter. So Penn State actually spray irrigates all of this wastewater that's generated at the university. And so they only have like a 6-hour holding capacity at Penn State. And so you have to spray irrigate out at this site. And so even when we had like this polar vortex back in 2014, OPP, our Office of Physical Plant guys, were out there like with blowtorches, making sure that the irrigation pivots were still able to run, because we have no other way of disposing of the university's wastewater. But it turns out there's about 14 vernal pools that are present at the field site. So we have both agricultural and forested lands that we do the spray irrigation at. And You can see very clearly one of our irrigation pivots is going right through this fancy vernal pool. So we're trying to understand what the effects of that irrigation activities there are having on these ponds. You can see our little monitoring equipment off to the side here. And so here's just some information about the site. It's sprayed every day. At about 8 of these laterals are turned on every day for 12 hours, and so they rotate which ones are on. You can't have one lateral turned on for more than once a week. So the soils are rested for at least a week after irrigation, and you can see how it looks like in the winter. It's actually quite beautiful. Go out to the site in the winter and see the effects of the spray irrigation and the ice sculptures, I guess, that it creates. But of course, we were trying to understand what the impacts are, not only to the vernal pools there, but that groundwater aquifer that we're recharging with that treated wastewater is actually our drinking water supply for the university. So trying to understand what the risks of doing these irrigation activities might be to our drinking water supply. And trying to understand how effective the soils, we have really deep soils there, thankfully. They're silt loam, Hagerstown silt loam, and trying to understand their, I guess, biogeochemical filtering capacity for these kinds of contaminants. So here's just a picture of the study site. Here's University Park, Penn State's main campus. The green stars are wastewater treatment plant, and the blue triangles are pumping station, and then all the wastewater comes out to the living filter. And those are the sites of our 3 vernal ponds that we decided to study for this work. And you can see down in these pictures where the ponds are relative to the nearest laterals, and so they're either right between 2 laterals, or in this one case, the lateral goes right through them. And so the equipment that we use to monitor, I thought since I have this field approach to research, it might be interesting to see the kinds of equipment that we use. And so we tend to use Campbell Scientific monitoring equipment. The sensors that we had were dissolved oxygen, electrical conductivity, pressure transducers so that we can measure the water level over time, pH, and an oxidation reduction probe. We wanted to understand the aerobic versus anaerobic conditions that might be present in the pond. As you can imagine, it's still water. They kind of get stagnant. Ask the students that do sampling out there. They don't always smell the best, and they have lots of duckweed and other interesting characteristics that sort of promote an anaerobic environment. And we measure rainfall and the irrigation activities with the tipping bucket rain gauge. As you can see kind of right here. We didn't have a really good place to put it, so just had to do the best we could. And then we used an ISCO automated sampler to pull water samples. And so the data that we generated when we were doing this study, we're continuing to do work out at these sites, but this is just the data that we have back from an earlier study that's now published. My first PhD student. We did an intensive sampling campaign during the time period from March to June, April to June of 2015, which basically corresponded to the time period for metamorphosis for wood frogs. And it turned out only one of our field sites actually had frogs in them that year. None of them had wood frogs, but one of them had spring peepers. But we were just trying to have some sort of relevant native species that we were designing our study around. So we selected wood frogs and just basically monitored some control sites and tried to figure out when they were hatching, and then we were able to design our sampling campaign around that time period. So we collected data from our sensor network every 15 minutes. We collected water samples daily. And then we also collected wastewater samples weekly from the irrigation laterals. So we had it set up that the irrigation laterals would be turned on once a week for each of the affected ponds. So these red lines are the irrigation depths, and the blue lines are the rainfall that we observed during that time period. So you'll notice for the water levels, of course, the most drastic changes were immediately following these irrigation activities. So they really impacted the hydro periods of these sites. And so here you can see the 3 ponds. And then in the blue line is the oxidation and reduction probe sensors measurements. And then in the dots are the different estrogens that we were monitoring. So the purple is estrone, as you would expect to see. That was the most commonly detected estrogen. And so what we saw was that only E1 was in the wastewater, yet we found 17 alpha estradiol, 17 beta estradiol, estriol, and phenyl estradiol, which is the control pill we're in the ponds. So we were hypothesizing that there might be additional sources. They were all higher than they were in the wastewater effluent concentrations as well. So we were trying to understand whether there might be transformation happening in the sediment, the vernal pools, desorption from suspended organic matter. It was just clear that we didn't have a full picture of where the estrogens were coming from that were winding up in the ponds. We did have elevated concentrations that persisted throughout our study period. So based on laboratory studies alone, you might expect, okay, the day or two following irrigation, maybe the hormones would be gone, but that's not what we saw at all. They persisted throughout the study period and appeared to either absorb and have additional presence because of additional sources within the ponds themselves. I mean, they've been irrigating out of the living filter since the early 1980s. Clearly, although estrogens are an emerging contaminant, they weren't just created 5 years ago. We've been poly-irrigating estrogens out there for as long as we've been doing irrigation activities. And so there could just be legacy effects like we think about for nitrogen and phosphorus that are also transferable to thinking about the fate and transport of these emerging contaminants in the ponds. So just some food for thought. Understanding your site history, I think, is really important in trying to understand what you're observing in the environment. And so we're continuing this work. We've actually expanded our vernal pool sites to include some sites in forested landscapes that aren't spray irrigated. But some sampling that we've done out of them has found caffeine actually at elevated concentrations, even relative to the spray irrigated sites. And so we think that septic tanks that may or may not be functioning so well could be really important sources of these contaminants in an environmental setting that you might not expect to see human wastewater indicators. And we're also deploying these POSIS samplers. I don't know how many of you in the room may have heard of them or used them, but they're basically these little membranes that will absorb any hydrophobic compounds, and so we're using them to understand what the fish exposures might be to act as integrators of the water quality over a month. We're deploying them for a month, and then we extract pharmaceutical compounds off of them to try to understand what's winding up on the membranes. And so we're also monitoring the groundwater wells that are present at the living filter. There's 14 of them. And again, we're trying to understand basically the biogeochemical filtering capacity of these soils to see if they're acting as a good filter or if we are getting transport of these contaminants all the way down into our groundwater wells, which are relatively new. We conducted a one-year sampling campaign across these 14 wells to understand that. And we also looked at the loads leaving the wastewater treatment plant. So these are some data from a Schreier honors student that I had who's actually an undergraduate student who did her senior thesis with me. So this is data she collected. And so she calculated fluxes of 7 different pharmaceutical compounds that were leaving the wastewater treatment plant, and then graphed that along with the flow rates leaving the wastewater treatment plant as well. And this dip, you'll notice, is winter break. That was kind of interesting. And then she actually calculated, based on all of that, the number of doses of these various pharmaceuticals that were getting spray irrigated on average every day out at the site. And so one of the interesting things that we found was that the spray irrigation activities are basically putting 800 cups of coffee back out to the living filter every day. So I thought that was kind of interesting. With 40,000-ish students at Penn State, 800 cups of coffee make it their way out to the living filter every day. And so then we also compared those wastewater treatment plant effluent concentrations to what we were seeing in the groundwater, and this is sort of averaged over seasons, so you can look at the effects of season on this, but basically we're finding that groundwater concentrations were at least 1 to 2 orders of magnitude lower than the wastewater treatment plant effluent. And so in general, soils appear to be acting effectively in reducing the presence of these contaminants. Although they did wind up there, it was really on the order of 1 to 10 nanograms or micrograms per liter instead of somewhere maybe even verging close to milligram per liter levels that were leaving the treatment plant. So interesting observations from our field work. And basically we were thinking that this is likely a good thing since that's ultimately our drinking water source. So we do now have a drinking water treatment plant that treats the water for Penn State, but that only came online within the past year and a half. Okay, so I guess the last part of this is trying to think about how we can move from fear to empowerment. And so, okay, awesome, thank you. And so basically what we wanted to do with this is try to understand what we could do to reduce the presence of the organic wastewater compounds that are in our wastewater. And if you look at a study that was done across the state sampling for these contaminants, you can see in orange that's the organic wastewater compounds that are the biggest contributor to the presence of emerging contaminants across the basin. And so basically what we wanted to do was figure out how we could use that information to make people feel like they could make a difference on this topic. And so we developed this emerging contaminants footprint calculator that the general public can use, and they just basically go through the products that they have at home, their personal care products and their household cleaning products, and enter it into this footprint tool, and it spits out basically an estimation of what their emerging contaminants footprint looks like. And then it gives you the top 10 products that are contributing to that footprint. And so what we're doing now is using that as part of an NSF-funded citizen science project to basically get people to think about what their own footprint is and understand their personal contribution to the presence of these contaminants in the environment, and then collect a surface water sample somewhere across the basin. It's kind of hard to see in this map. I didn't have a better map to put on here. But all of these little white numbers are up to 60 sites that we've selected for various reasons, because they're upstream or downstream of the wastewater treatment plant or various land uses. And then we send the citizen scientists that have volunteered for this project a sampling kit. They go out, pull a sample from their site. We have them all back now. We're analyzing them. And later this summer, we're going to have focus groups where we basically discuss how to link what you see in that footprint tool to what you're seeing in your water sample, and understand contamination across the presence of these contaminants across the basin, and then what people would like to see done about it. So we're hoping that that will sort of have us set an agenda for the future research that our group does in response to the stakeholder and citizen science input into those research questions. So of course, I have a whole suite of people I need to thank who have contributed to all of this, including our funding sources. But I think that citizen science project is one of the most exciting things we have going on in our lab group right now. So hopefully some of you have heard about it, and maybe even are volunteering as part of it. So thank you for all of your help. I'm happy to be here today. And if you want access to any of the calculator or my research website, they're all here. Great, thank you.
So I wanted to give Greg Allen a few minutes. Greg's focused on PCBs and talk a little bit about the science and modeling needs for PCBs, and then we'll open it up to the audience.
Okay. Good morning. Again.
Good morning.
Yes. Yes, thank you. How's the volume? Can you hear me?
Yeah.
Yeah? Okay, great. So first of all, thanks to Bill and Scott for their leadership in bringing these toxic contaminant related issues before the research and science community. PCBs, as Scott mentioned, are the contaminants that drive most of the fish consumption advisories in the tidal waters of the bay. In the non-tidal waters, it shifts over to mercury, but PCBs drive most of the impairments in tidal waters. And even though they were banned in 1979 from new uses, they remain among the champions of persistence, bioaccumulation, and toxicity. Our best professional judgment is that on a mass balance sort of perspective, the PCBs are being metabolized in the system more quickly than they're being loaded in. But the slope of that line, we think, is very shallow. And so the Bay program and its toxic contaminant work group and the partners in the Bay program are coming together to try to increase the slope of that line. It's really what our objective is here, is try to find ways to increase the rate of removing PCBs from the system, and specifically from the fish. According to some documentation that's on Virginia DEQ's website, all of us have PCBs in our bodies. It's highly likely that if you've ever eaten fish, that you have PCBs in your body right now. And we think that that is a situation that is untenable and really deserves a lot of the focus from the Bay Program partnership. We're also hopeful that as we get past the midpoint assessment of the nutrient and sediment TMDL, that we'll see some shifts of resources to focus on some of these issues a little bit more. Specifically for PCBs, 3 areas that we need ongoing support from the science community, and they are, as you might guess, in modeling, monitoring, and understanding the management actions that are most effective in reducing the bioavailability of PCBs. So in modeling, we have a fairly dynamic modeling situation for PCBs, but they're localized. The extensive impairments and TMDLs that are in place for PCBs are localized. So we've got the Potomac tidal TMDL for PCBs that has a fairly well-developed model. But there are other places where models are in development now. For example, on the James River, and also the Susquehanna in Maryland, the lower Susquehanna, and the Conowingo Pool we'll have a PCB TMDL coming online. And in each of those cases, there is models that are being developed, support from VIMS, in the cases of the James and the Susquehanna models. But we need to continue to know that we're improving those models, particularly with regard to how the different jurisdictions are cooperating on the PCB TMDLs and using the models to inform that cooperation. PCBs are not unlike nutrients in many ways. These impairments are across the watershed, and the interstate loading of PCBs is the same as it is with nutrients. They travel across state lines. We need our models to continue to improve, and we need our jurisdiction partners to be using those models to collaborate on reducing loads. We need the science community to support us on monitoring. The models aren't worth much if we're not able to feed them with valid data. There's a problem with PCBs and monitoring. We need to be able to see very low concentrations of PCBs. There are many diffuse sources, and to be able to identify them and track them down and find them and remediate them, we need low-level detection limit data. And currently, the method that gets us that data is method 1668, high-resolution mass spec, and it's very expensive. Right now, it's about $700 or $800 per sample in the marketplace right now. So we need to continue to have our analytical chemists help us figure out ways to reduce those costs. There are other monitoring methods that are appropriate in certain cases, and again, we need our scientists and our analytical chemists to help us know that we're matching the right method to the right data need, and including how we get the low detection data that's really critical to modeling and finding sources and getting them remediated and reduced. We also have a gap in the air deposition for our PCB models. We need more monitoring to understand how that's part of the flux of PCBs in the system. Right now we're running off of information we have from the late 90s. And that remains a data gap and a need in our monitoring and modeling activities. We don't have great trending data right now. There is no comprehensive monitoring network for PCBs the way that there is for nutrients. It's spotty. And we need to figure out how to get the data that we need to be able to know whether our TMDLs, our PCB TMDLs, are being effective, getting the job done, and that's the trending information that we need. Lastly, science need for understanding the management actions that are effective in reducing PCBs. That's both from the BMPs, in particular urban BMPs, and from wastewater treatment plants. In the Delaware estuary, there's been great progress in reducing PCBs. We know we can do this because our partners to the north in the Delaware estuary have made fantastic progress. They made a lot of that progress by reducing wastewater treatment loads of PCBs. It's a small concentration, but it's a lot of volume. It adds up to a significant part of the load, and they made some very good progress there. So we will have a report. We're doing a literature review right now, and there will be a report about what we know happens when plants are upgraded to ENR with regard to PCB reductions. And I can tell you that the preliminary result is that there is a reduction. Now the question is, where are the PCBs going? And we're basically partitioning over to the sediments in the wastewater treatment plant. We need to understand more about how those sediments are, those solids are disposed of, and what it means about PCB cycling through the system. Wastewater is important in PCB TMDLs. So those are a few examples of our ongoing science needs in modeling, monitoring, and understanding the management actions that are most effective. one more point I want to make is that there is a potential exciting opportunity for the research community to come together to support this critically important problem of PCBs in fish. We are proposing really the agreement we have right now is to explore the possibility of establishing a PCB consortium. The reason we feel this is needed is because there is no collaboration and coordination. There's no entity right now that is commensurate to the scale of the problem. We have PCBs across the watershed, and there really is no place right now where both the science community and the managers are coming together to cooperate on this issue in a way that we think is befitting of the importance of the problem. So we don't envision a Bay-wide TMDL for PCBs, and in the meantime, we don't really have a way for the science community and the regulators to work together on this. And so we have agreement from our Bay Program Management Board to explore this concept and bring back to them some information, hopefully leading to a decision as to whether the Bay Program Partnership would support this effort. And the consortium, again, would be a combination of science issues and making sure that the folks like Upal Ghosh, who is at UMBC and doing some fantastic research about remedial technologies for PCBs, are working closely with the managers and regulators and making sure that the information is being shared and that the states are cooperating on the interstate issues around PCB transport and their TMDLs. So we will have more information on this over the coming year. But again, I think it would be a great way for this group of folks, really, and those among you that have an interest and a passion around this issue to coalesce and come together to support the regulatory community to figure out how to increase the slope of the line and get PCBs out of the fish, and all of us and all of the stakeholders and the users of the resource of the bay at less risk because there's lower concentrations of PCBs in the fish. So stay tuned for that, and maybe some of you that are here today will become the core, the guiding coalition within the PCB consortium that could support the regulatory community on PCB reductions. So that's comments I wanted to add.
Right. Thanks, Greg. That was really useful. Round out what we're trying to accomplish. So why don't we open it up to you all? You've been patient, so some questions from the audience or issues you want to bring up. Yes.
Hi, I have a question for Lee, actually, on what were the macrolides you were looking at in your antibiotics?
Yeah, so we're pretty much measuring all of the macrolides right now, the full class, maybe with the exception of one or two of the more recent ones. I can give you all those details afterwards, but pretty much we're looking at all the macrolides, all of the tetracyclines, about half of the sulfonamides, and about half of the fluorochemicals.
Okay, great, thank you.
Others?
A question for Vicki. When you were talking about work in Pennsylvania and you showed some correlations between intersects and different factors, there was one, I think the significance wasn't that high, but there was a strong negative correlation between intersects and wastewater treatment plants. I was curious about that.
And we have seen that before. And for some reason, it seems that smallmouth, and that was smallmouth bass work, that smallmouth bass are responding to something in ag. When we look at the white sucker, as I mentioned, they have vitellogen and they don't have intersects. There we see a different pattern. They have higher vitellogen downstream of wastewater treatment plants. That's not what we see with smallmouth bass. So our theory, and right now it's just a theory, is that wastewater treatment plants may be taking out some of the things that smallmouth bass are actually responding to in agricultural runoff.
Yes.
This is for Lee. The results that you showed for the sunscreen compounds on the lower eastern shore and in Tangier Sound were kind of interesting because there's just not that many people there. Can you comment a little bit on the sort of the reactivity of those compounds, and then also maybe wildly speculate on why you're seeing those results?
Yeah. So in terms of the activity of the compounds, I'm not going to speculate. I'm not a toxicologist, and so I think there's really not been very many toxicity studies for the UV filters, but they have particular chemical moieties that have been shown to exhibit toxic effects in other organisms. The one that we're really concerned with is that phenol moiety. And so one of the things that's a little bit different about the UV filters compared to the other molecules that we work with is that there's not like a conserved chemical structure. And so in pharmacology, we would call that like the pharmacophore, and then you basically change out different functional groups. The UV filters are a little bit different, but again, they have these moieties that can exhibit estrogenic effects, and so I think that's something that bears more work, in the lab as well as the fields to just really build out that toxicity. Because at this point, we don't know. Like even the regulations in Hawaii, I would say there's not a lot of data showing toxic effects in corals. There's a couple of studies, and they just kind of really blew up and got this conversation going. And I think that really led to these regulations. But there's really not a lot known about toxicity in general.
Just to follow up, how long do they persist in the environment? And sort of what's the main mechanism of degradation?
Yeah. So that's something that we're looking at right now. Actually, these chemicals are meant to absorb light. They do a very good job at absorbing light and then undergoing either, in these cases, both direct photolysis or indirect photolysis. We're looking at many of them in detail right now in the lab to get a better sense of their photolytic degradation mechanisms. But as I mentioned, they're also quite hydrophobic. The log KOWs can range from about 3 up until about 7. And so they are very much expected to partition into organic matter and organic sediments, as well as organisms. And so I think, to come back to one of your earlier questions about how they got there, that's definitely a question I want to have conversations with people. In some of our brief conversations so far with other modelers, I understand that there's very complex transport patterns within the bay. And so even if we're seeing some of these things showing up in these more isolated corners, it's not necessarily kind of our first assumption that they're coming from directly upstream. They may be coming from across the bay, or they may be coming all the way down the bay. We just don't know that yet. And so if other people do know that and transport problem or transport questions in the bay, we'd love to have that conversation. Thanks.
Blue shirt.
So I actually work in another research area which is focused on exposure to chemicals in the indoor environment. And so it's like a very similar problem. So you've got thousands of chemicals, thousands of sources, maybe a few exposure pathways, thousands of different toxicity mechanisms. It's an impossibly complicated problem. And so then the question is, well, how can you sort of simplify this problem a little bit? So you can't measure everything, you can't model everything. So maybe what you can do is kind of come up with a simple exposure model, not a very complicated one, a sort of simple exposure model. It's like a screening level exposure model that has the main the main mechanistic sort of components, but it doesn't go into all the detail. And then you need a simple estimate of toxicity. So maybe you know that EPA has this new program called ToxCast. It's like a high throughput program to rapidly come up with estimates of toxicity to a whole huge range of chemicals. So then the idea is if you have a quick estimate of exposure and a quick estimate of toxicity, you can put them together and get risk. So now you've got a quick estimate of risk. And if you do this, you find that the risks vary over maybe 10 orders of magnitude. If you consider all the chemicals and all the sources, 10 orders of magnitude. So it's not a factor of 10 orders of magnitude. So there are huge differences in toxicity. So if you could do this in a screening way, then you could just focus on the top one, you know, the ones at the highest risk, where you have the biggest exposure and the biggest toxicity. And then maybe, because it's such a complicated thing, then you kind of focus on those ones. So is it possible, do you think, to do something similar for the problem that you're addressing here?
Great question.
I'll just comment a little bit because some of that was actually done at some of the sites, not here in the Chesapeake, but in the Great Lakes where we were looking at fish at areas of concern. And a group from EPA took a lot of the ToxPass methodology and came up with risk. And what was interesting is that they looked at the risk for intersects as well as vitellogenin induction. And at least 80% of the sites where we found intersects and vitellogenin induction in bass, they were predicting that there was no risk or low risk. So again, I think it could be a good first step to find some of the contaminants, but it's not going to always work, I guess, is the...
I mean, there'll be big uncertainty, you know, there's big uncertainty,
right.
but if you look at the overall problem, there are huge uncertainties, you know, in that overall problem. And so, you know, you could live with being wrong by a factor of 5, you know, no problem, because it doesn't matter, you know. So anyway, I think it's a worthwhile thing to consider. You're not going to solve the whole problem by doing that, but I also feel like you're not going to solve it in the way that, you know, the current approach. So, you know, it's an impossibly complicated problem. There's no hope of solving it, you know, in this kind of, I don't know. Anyway, that's my sort of,
Okay.
Maybe if I can just make one more point. So then, you know, I'm interested in the indoor environment. And wow, exposures there are high, you know, from a human exposure point of view, way higher than exposure to what you get from exposure to waters. And so, you know, do you ever consider sort of human exposure in other environments to the same chemicals, like PCBs? You know, there's quite a lot of human exposure to PCBs coming from the indoor environment. So if you kind of compare, you know, between the sort of natural environment and fish and the indoor environment, say, okay, the big problem is in the indoor environment, you know, not. Maybe that would also be a useful thing to kind of go after the high risk problem.
We haven't, but we're very interested in some of those questions, especially with respect to sunscreens. And, you know, I think in the past, most of the sunscreens were kind of lotion-based. You know, these days I get the sense that probably most people are using the aerosolized ones. And so we have a lot of ideas there and questions there that we're interested in. So if that's a common interest, I'd love to have a conversation.
All right, we're up against our break, so if you have some questions, I know our speakers will be around, so try to grab them during the break or at lunch. Otherwise, a round of applause for the panelists.